Hi, my name is Santosh Mishra and I am Assistant Professor of Neuroscience at the Department of Molecular Biomedical Sciences at the College of Veterinary Medicine. So my lab research focus is pain and itch. We are interested in these two sensations because both these sensations are known as a major aversive stimuli that are encountered in our day-to-day life. Though unpleasant, but these sensations play an important protective role in alerting us to possible dangers or irritants that could cause harm. So when disease conditions like osteoarthritis or skin disease like atopic dermatitis and psoriasis, which afflict our body, the pain and itch sensation can become chronic and even become more worse. Outside of these non-steroidal anti-inflammatory drugs like NSAID, which often cause serious side effects, there are only few options to treat these pain and itch sensation. And the reason is that there is no neural circuit or mechanism known for these pain and itch sensation in these disease condition. So it is important for us to understand the neural circuit and the mechanism behind these nociceptive stimuli in both healthy and disease condition. So what do we know about pain and itch and about how our skin detects and our brain processes these stimuli? Pain and itch are detected by neurons whose cell bodies are actually located in the dorsal root ganglia. So these axons split bidirectionally, one going into the skin and the other is going into the spinal cord, where from there it goes to the central nervous system for processing. So historically, it was known that the DRG sensory neurons are polymodal. So which means that the neurons express several different kinds of receptors to detect and transmit these signals to the spinal cord and via these neurotransmitters. So recently we and others have identified that these neurons in the DRG are label line for detecting these nociceptive stimuli. So what does a label line means in terms of detecting these nociceptive signals? It means that the subset of neurons in the dorsal root ganglia express these selective receptors to perform a particular functions. So neurons in the DRG are heterogeneous based on the expression of these receptors. So for instance, a transient receptor potential family TRPV1 is involved in sensing heat, whereas another gene from the same family, TRPM8, is involved in detecting cold. So both of these receptors were reported to be expressed in two different subset of neurons and to perform independent function. So most recently, what we identified using these RNA sequencing and microarray, that a small subset of neurons within these TRPV1 population express a neurotransmitter called NPPB in the DRG, which is actually a neurotransmitter connects the DRG sensory neurons to the central nervous system in the spinal cord. So we further show that these NPPB is involved in itch transmission and further identified three stations from the DRG to the spinal cord that are involved in propagation of itch signaling. So our lab research focus is to expand our current knowledge of acute sensation of itch and pain at the DRG and at the spinal cord level to unravel the neural mechanism behind these two major diseases, osteoarthritis and allergic skin disease, for example, like atopic dermatitis. So our very long-term goal is to advance our research into the brain to dissect how information from the DRG is mapped, interpreted, and converted in the various brain itch and pain centers. In summary, the overarching goals of our study is to elucidate a potential neural circuit for itch and pain that could possibly be used as a target to alleviate these two obnoxious sensations.